Hello, everyone. Thank you, Rafa, for this kind introduction and for inviting me. It's really, I'm delighted to be here with you today and share some of the recent work that we've done in the lab and with collaborators. So with that, let me begin. The code and data sets and projects that I will highlight today are also all available from our lab website. So if you go there, you will find pointers to papers and data sets and code bases. And I will also provide pointers throughout the talk. Happy to take any questions during the talk if there are any pressing questions regarding terminology or that things that require clarifications. And I also hope to leave some time at the end for a robust discussion. So with that, let's begin. So the state of a person or individual is being now described with increasing precision, incorporating various modalities that range from genetic code to molecular interactions, therapeutics, and all the way to the environment. And so that really gives rise to these multi-scale complex biological systems where it is the case that no individual data point is independent of each other, but they really depend on each other, they interact with each other, and kind of that kind of complexity and interdependence is pervasive in biology at all levels of biological organization. The challenge then becomes how to computationally operationalize these data to make them amenable to learning and human decision-making, as well as to derive hypotheses that can guide biomedical discovery. And so at the core, the question that becomes, well, how can we infuse structure about those systems and prior knowledge into algorithms that we're developing? And so the algorithmic paradigm that has enabled that over the last few years has really been driven by the success and advances that we have seen in the broader area of graph representation learning or machine learning for graphs or interconnected data. That has led to many areas of success, including those in the area of therapeutic innovation and drug discovery, knowledge graphs and ontologies, single-cell biology, multi-omics data integration, and others. And so today I will highlight some of the work that we have done, as well as in relationship to what has been done before, that touches areas from molecular graphs and modeling of protein interaction networks, to ontologies of cellular systems, to knowledge graphs, and also single-cell gene interaction networks. So the plan for today is to cover three broad topics. First, I will talk about the advances in transfer learning and graph neural networks and what those advances have enabled us to tackle two exciting problems. One is finding cures for emerging pathogens that we have never seen before, and also classifying novel cell types in large single-cell data sets. In the second part of the talk, I will focus from the methodological standpoint on learning with knowledge graphs and explainable AI, and I will describe how advances in those fields can help identify therapeutic opportunities for diseases and prioritize combinations of drugs for safety. And finally, I will conclude with a topic that I think it's incredibly important and often neglected in research, that relates to the question of how to create flagship data sets that can really facilitate both algorithmic and scientific innovation in the particular area of therapeutics that I'm very excited about. So with that, let's begin with the first topic. So motivation for this work is really a question that I've been asking myself for a while. And I really like that question because it often guides me towards coming up with a very crisp answer about what is the core aspects of biomedical data that makes biomedical data unique and therefore prevents us from simply reusing some of the off-the-shelf machine learning algorithms. So for this part of the talk, the motivation is the observation that for many problems at the frontier of biological discovery, we have very little labeled data available. And so this is an important observation because recent breakthroughs in machine learning have depended on training very large models with a large number of parameters and doing so with a large number of labeled data available. So that has led to tremendous successes in computer vision that was fueled by a large flagship data set, ImageNet, with millions of labeled images, as well as similar data sets in natural language processing. Those very large data sets have enabled machine learning engineers to build and train models with many parameters that then led to broad generalization. The problem is that for many problems that we are very excited in biology and medicine, there are no such very large labeled data available. So that really creates an urgent need for having models that are labeled efficient, that can work in situations where we have only a small number of annotated examples. And so there are many domains where this is a real problem. For example, when we are developing algorithms that can assist in the development of novel drugs, that becomes an issue since the labeled information for truly novel drugs in development is scarce. Modeling emerging pathogens or rare diseases, or modeling hard to diagnose patients are all problems that typically come with very few limited label data, and therefore prevents training standard supervised framework. Visually, the way you can think of this problem is that what prevailing methods assume is that what we have the settings shown on the left part of the slide. So we have these rich interconnected data sets where most of the nodes in that data sets, most of entities, are already somewhat well characterized. Their labels, here represented by nodes of different colors, are already available. And then the question becomes, well, we can leverage all that label data to predict labels for remaining entities. But what happens in real world is that we more often see the situation shown in the right part of the slide. We have these vast regions of gray space where most of entities, or many of those, are completely uncharacterized. They're this dark gray matter that we need to be able to prioritize, identify those that have a certain phenotype, for example, of interest. So to address this problem today, I will outline an approach, a general framework that allows us to tackle these particular hard settings where limited label data are available. And then I will show how we can use it for biomedical discovery. So this broad area of research is known as few-shot learning or meta-learning. So one slide of background about few-shot learning is next. So the way to think of this problem is the following. So few-shot learning is a particular sub-area of a broader field, which is known as meta-learning. And it's concerned with the situation where we have a small number of labeled examples. So in particular, what does that mean? So the terminology that is used in this area is refers to the number of labeled examples as shots. So the example that I'm showing here on this slide is an example of two-shot, three-way few-shot learning. So what does that mean? That means that we have a situation where we need to train a classifier, in this case, an image classifier. And that image classifier will need to distinguish between three classes. And for each class, it has just two shots, two labeled examples. And so the way few-shot learning makes it possible to train a classifier that can distinguish between images of different classes, having seen just two images for each class, is through a specific kind of specific training strategy that has two phases. That training strategy has the first phase, which is meta-training phase. And during that time, we will take the classifier that we want to make generalized to these challenging problems with limited label data, and we will expose that classifier to very hard learning settings. We will challenge that classifier to perform well under limited settings. What that practically means is that we define a number of small training tasks, and each training task is designed to be really hard. So training task number one, we ask the classifier to take these two labeled examples for three classes. So we have two cats and two sheep and another and kind of wild boars. And given all the data, then the classifier needs to be able to do the best it can to then classify images that belong to these three classes. And so the model might not perform well after it sees just one such training task. So what we will do is the knowledge that it acquired on training task number one will be transferred to training task number two. We will initialize the model for training task number two with what it has learned in training task number one and repeat that many, many times. And each time the model will become better and better because it was exposed to these kind of hard challenges. And so this is important because at test time, the second stage of few-shot learning, when we actually need to build a classifier, in this example, duck versus dolphin versus chicken classifier, having only 2 labeled examples for each of those 3 classes, we can actually do that because the model was trained in such a way that it can very quickly adapt to a new class. We trained it in that such regime. Okay, so what we've been developing over the last few years are algorithms that can make few-shot learning really practically useful and also theoretically well-grounded. So the algorithm that I will highlight is called GMeta. It has been published at NeurIPS now a year and a half ago, and kind of it operationalized this principle of designing challenges for a classifier and does so for graphs. So in this particular case, we have a similar regime. During the meta training phase, we will train a graph classifier. In our case, that would be graph neural network that will only see a small number of labeled nodes for a given label. So in this case, in training task number 1, those are just 2 labeled examples of light gray shade, light yellow shade, and then just 2 labeled examples of yellow and dark yellow shades each. And given only that information, the model will learn how to discriminate between these three labels. And it will do that and will expose the model to many training tasks during meta training phase, and each time transfer the knowledge that the model has learned on all the tasks so far when solving a new task. And so that will be helpful because then during the test time, when we need to now build a classifier for a new type of labels that have never been seen before, in this case, those would be labels of blue with blue shades, then the classifier can do that, having only 2 labeled examples for each label. Okay, so how can this be done? How is that possible? So the key idea of GMeta is that it relies on a principle that we refer to as local subgraphs. So the intuition is that because the amount of labeled data, the amount of nodes that are labeled is very small, that information alone simply will not be sufficient to train robust classifiers. So what GMeta does here is that in addition to looking at those small number of labeled examples, it also looks in the local neighborhood around those labeled examples. And those local neighborhoods are local subgraphs. And essentially for each of those local subgraphs, GMeta specifies a nonlinear mapping function f that takes the central node of the local neighborhood and its connectivity structure, maps it into some d-dimensional and compact embedding space, such that for 2 local neighborhoods, for 2 local subgraphs, if they have similar topological structure, in addition to having similar labels, they also need to have similar topological structure, and that information is what is leveraged by GMeta. They will be embedded close together in the embedding space. And so that structure of subgraphs serves then as a signature function that essentially when we go from one training task to another task, each time that function f that describes what are the topological signatures of those local subgraphs is transferred from one task to the other. So when we expose the model to a new training task, we initialize that parameters for that new training task with what the model has learned before. And so over time, the model becomes better and better and better. So why this idea of using local subgraphs, not only it is theoretically well-grounded, because what we can show is that the influence of a node and its particular label, the influence of a node on the target node that we want to make a prediction for and whose label is unknown, the influence of other nodes decays as we go further away from the target node. And so what that means is that it's possible to show that if we focus our models into effective exploration of local subgraphs around those target nodes that we want to make prediction for, we can capture all the relevant information. So this idea of local subgraphs is then useful not only because it enables broader generalization, but it also can capture near the same feature information as if modeling the entire graphs, which makes the approach very, very scalable. Okay, so this might seem a bit abstract. So to make things a bit more practical, let me share with you two studies where this idea of few-shot learning on graphs have been practically used. So first I will highlight a study where we used GMeta and few-shot learning on graphs to identify therapeutic opportunities for an emerging pathogen. And second, I will talk about identifying novel cell types from heterogeneous single-cell experiments. So for this first application, the motivation for it originates from the need for rapid, effective, and safe therapeutics developed at incredible speed to tackle some of the challenges that that COVID pandemic has brought. And so that the observation was that the traditional iterative development and experimental clinical testing of approval of new drugs is not feasible in situations where a public health emergency requires response that is very fast in identifying novel effective drugs, because the design of a drug from scratch on average takes between 11 and 16 years. And so that's certainly not feasible in the situations that we were at. So the question was then, well, is it possible to compress years of work into months or even weeks in some situations through the use of advanced ML models and new named data sources and automation? And in particular, can we do that by looking at drugs that are already on the market or in later stages of clinical trials and repurpose them for new users? So the study that I will mention, it took place, the central part of it took place between March 23rd, 2020 and June 15. And during that span of just 3 months, we were able to collect the data and then practically implement these few-shot learning strategies, identify promising therapeutic opportunities, and then with collaborators engage in experimental verification of those predictions. So how did we do that? First, we built a data set, and so the data set had the following structure. It consisted of the human protein-protein interaction graph. So this is a network where nodes are protein coding genes, and edges between them indicate physical interactions between proteins. And on top of that protein-protein interaction graph, we have information about approved drugs and their mechanism of action in the forms of proteins that each of those approved drugs in the US targets. And then also information about diseases and proteins that are known to be perturbed in each disease. And so having that information for thousands of different diseases and thousands of different drugs, we were then able to study the relationship between drug targets and these proteins perturbed in diseases in the context of what drugs are approved or are known to be efficacious and safe for certain diseases. So given that information, so the human protein interaction graph, information about drugs and their protein targets, information about diseases and their associated proteins, and the knowledge about what drug already treats a disease, our goal was that given these approved disease treatments, can we identify and prioritize treatments for COVID-19 in a manner that is known as zero-shot manner. So why is this so challenging? Because there were no examples of successful treatments for COVID-19. So it was really like, can we prioritize these drugs, but we don't have any successful treatment for that emerging pathogen. So no label. That's why this is a zero-shot learning problem. So the way we set it up is the following. So we asked our deep graph ML model, we trained it in the following regime. So during the meta training phase, we designed a large number of training tasks. Each of those tasks was designed for a different disease. So in training task 1, we asked the model to learn how to distinguish between drugs that treat disease 1 versus drugs that are not treat disease 1. And once the model did that, we took its signature functions to initialize the model for training test number 2, when the model was again asked to distinguish between asked to continue optimize its parameters, to distinguish between drugs that are indicated used to treat the disease 2 versus drugs that are not indicated for disease 2. And so we did that for all of the diseases with known treatments. And so after that was done, in the meta testing phase, we asked the model in a zero-shot manner to prioritize drugs based on this COVID-19 emerging pathogen that had no examples of successful treatments, with the exception that we know what are the proteins that the virus binds to. And so the model was able to produce a priority list of drugs such that at the top of that priority list were those that the meta approach thought as of being most promising. So the first test that we did at that time was, because there were no examples of successful treatments, how could we even estimate how good that priority list is? So we did a simple study and a lot of those kind of simple checks, but here I'm just showing one example where we looked at drugs that were at the clinical trial clinical trials at that time, and we correlated priority list of drugs produced by GMeta to list of drugs ranked by in how many trials they are currently considered. And so we compared that against various standard supervised learning frameworks and their predictions. And what we found is that GMeta consistently was able to outperform many of those standard diffusion-based methods based on network proximity, graph kernels, and others. So that's a very weak way of evaluation, because we certainly know that the fact that a given compound is considered for being a therapeutic in clinical trial, this of course does not mean that it actually works. And so, but it convinced us that these predictions might be worthwhile and might be promising enough to consider them further. So what we did is we teamed up with National Emerging Infectious Disease Laboratories at Boston University. This is a level 4 laboratory where they can work with live virus. And we shared with them some top predicted drugs by the model. And what they did is they took those predicted list, and they used those predicted lists to inform their experimental screening. First in African monkey, via E6 cells, which was a model organism for this pathogen. And then later on, they also did experiments in human cells. So without going too much in details, the results showed that by screening top-ranked drugs in human cells, we obtain a near 62% success rate, meaning that 62% of compounds that were predicted were shown in experiments to inhibit growth of virus in different conditions and at different dosage levels. In contrast, what would be possible to obtain if that experimental screening would not be guided by any prediction tool, the hit rate would be merely 0.8% hit rate. And so this is an order of magnitude higher hit rate among those top-ranked drugs, which was very exciting to us. Furthermore, what we were interested in seeing is that among those drugs that were predicted to inhibit viral growth and experimentally verified that they actually do that, 67 out of 77 drugs that successfully reduced viral infections were those drugs that did not directly bind to proteins that the virus binds to. And so why is this significant? This is significant because it means that standard or more classical methods for drug repurposing that are based on molecular docking simply would not be able to identify those drugs, because they can only look at those effects that are the result of direct interactions between the targets and the drug. But there are this group of drugs that behave in a way that is not consistent with that characterization. They actually have these strong network effects, which we now refer to those groups of drugs as network drugs. And it's really shows that these graph-based machine learning models, not only they are accurate and useful and we like them, but they also enable can lead to discoveries that would not be possible to obtain with other techniques. Okay, so the important part here where really the few-shot learning algorithm was crucial was because it would not be possible to otherwise prioritize candidate therapeutics for this emerging pathogen because we didn't have any example, any reliable example of efficacious therapeutics for COVID-19 for emerging pathogen at that point. So it was a kind of this zero-shot learning problem that we were still able to tackle with ML algorithms. And so let me show you another example where the similar idea was used in a very different context. This time we're interested in looking at heterogeneous single cell experiments that come from different tissues and then see whether we can design a tool that not only can annotate cells for their cell types, but in particular can identify there are groups of cells that might constitute a possible novel cell types that and how to characterize that group of cells. So the project we developed here is called MARS, and the key idea of MARS is the following. MARS assumes that a set of input, that it takes as input a set of heterogeneous annotated experiments. So heterogeneity here comes primarily in the form of having single cell experiments from different tissues. And it assumes that, well, we have some number of those heterogeneous experiments that are annotated with their cell types. And so the output of MARS is that given those set of heterogeneous annotated experiments, we want to annotate a new, completely a new experiment that is not annotated at all. For example, for a new tissue like brain tissue that has not been seen before or modeled before in training time, and we want to do that even if that new experiment does not have any cell type in common with experiments that we have seen before. And so the approach that MARS takes is the following. So the idea is that we'll take this set of heterogeneous annotated experiments, and MARS will learn what and optimize for a low-dimensional compact embedding space that will project all cells in that input data set, both annotated and annotated, into that shared embedding space. And so this will be done during this process of meta training. So we refer to this collective data set as a meta data set. And so learning this shared embedding space is done such that it requires learning some parameterized function f, such that two cells from the same cell type will be embedded close together in that embedding space, and cells from different cell types will be embedded far away from each other in the embedding space. The crucial part here is, again, this question of what is the transfer mechanism that will allow us to go from one training task or from one cell type to the other. And while we have seen that before, that in the context of GMeta, those were these local subgraph signature functions, here we also have such a mechanism that we now call cell type landmarks or cell type prototypes. So MARS relies on cell type prototypes. Those cell type prototypes or landmarks are learned jointly with embeddings for cells. And so they are learned such that we optimize this shared embedding space for three objectives. These objectives are the following. Within each annotated experiment, we want to minimize the distance between the embedding of a cell and the embedding of a landmark representing the cell type that that cell belongs to. And so we can do that because that is for annotated experiments, and annotated experiments mean that we have cells and their cell type annotations. If within each unannotated experiment, where we don't have the ground truth label of a cell in terms of its cell type, therefore we don't have ground truth landmark, we will still strive in an unsupervised manner to minimize the distance between embedding of cell and the distance to its closest landmark. So we still want to, the model has a desire to embed a cell to a landmark, close to one landmark. And we also, of course, we don't want our landmarks to be to kind of be grouped very close together, which would be kind of a trivial solution. Which, because of that, within each experiment, we want to maximize the distance between landmarks. So we want to have a minimal number of landmarks to cover as large space as possible. So what learning then both embeddings of landmarks, these cell type signatures of cell types and embeddings of individual cells, then allows us to allows Mars to achieve to accurately annotate cells, both in this more standard setting where we need to annotate cell types by looking at a new tissue. And so I'm showing here on the left part of the slide a comparison of Mars to existing methods for cell type identification. And these results are summarized across 21 tissues in tabula muris, which is a single cell effort at Chan Zuckerberg Biohub. And so here in this case, Mars was trained in a leave one tissue out manner. So we repeated an experiment 21 times. Each time there was one tissue that was left out, and the remaining 20 tissues were used to train a Mars model, and then the performance was estimated on that left out tissue. And so what we can see in this plot is where on the x-axis are different algorithms, and on the y-axis is performance metric. This is adjusted trend index. Higher value indicates better performance. We can see that in this setup where we test the ability of the model to generalize to a new tissue, Mars can do that much better than existing work. And this is possible because of those cell type landmarks. What is even more exciting is that in this plot, I believe on the right part of the slide, where we took Mars and the second best performing baseline model, which was similar in this experiment, and we calculated performance of Mars and similar measured as F1 score. Again, higher value indicates better performance, and we did that as a function of how many cells does a particular cell type have. We were interested in seeing whether the model can perform well in situations where cell types are small and there are very few cells sampled for a given cell type. And we have seen it that in particular in this hard regime, with their small number of cells in a cell type, and they're therefore harder to recognize those cell types by algorithms, Mars can perform much better than existing methods. In addition to improved performance of the model, I think that particularly exciting capability that Mars has is that it can automatically generate interpretable or human readable names for new cell types. And so why is that important? Well, that's important because if you have an algorithm that says, here are these groups, here's a group of cells, a set of cells that were measured and that somehow do not fit into distribution of cells and cell type that have been seen before. A natural question that emerges is, well, what is then common to those cells in that group of cells? How can we characterize that group of cells? And so what Mars can do, by relying again on cell type landmarks, it can provide human interpretable names for these for the cell types that it has discovered as possibly being candidate new cell types. And so how does that work? It has two important steps. And so first is for each new cell type that we want to name, what Mars will do, Mars will calculate the distance in the embedding space between each cell in that new candidate cell type and all of the landmarks that the Mars has in the embedding space. And then for each landmark, Mars will output what is the probability that this new cell type should receive the same name as the name of that the landmark represents. And so this produces then outputs such as shown here in this example below, where we can see that we can visualize, we can see in the central is the joint embedding space learned by Mars, and these blue pentagons indicate positions of landmarks from annotated experiments. And so when a group of cells that we might highlight as possible new cell types, new cell type would be highlighted, then Mars would produce this kind of distributional plot that would say, well, how likely it is that this group of cells might be each should receive the same name as those cell types that are already known. And so here is one example of the cells that we selected were actually macrophages. And so Mars has really correctly identified that with 87.9% confidence, this group of cells should get the name macrophages. And then with some 3.9%, this would be then myeloid cells and neutrophils and so on. And so it can do that for any group of cells that one would think of as being possible new candidate cell types. And so these are really human readable names that are exciting. Okay, so with that, I hope that kind of it was clear how in this particular case, the role of few-shot learning was in identifying mechanisms of transferring knowledge from one experiment to the other, and that transferring that information was encoded in embeddings of landmarks that represented cell types. And once we learned that, though in the embedding spaces, those landmarks can really be effectively utilized for classifying new cell types, for naming cell types, and also for using the method in hard settings, such as leave one tissue out settings or leave one cell type out settings. Okay, so in the second part of the talk, I will change the direction a bit. And so so far we talked primarily about few-shot learning and how to train machine learning models in the context where limited label data are available. And so in the second part, I will focus more on heterogeneous data representations and knowledge graphs in particular, and how we can leverage and learn in those settings. So the motivating example and biomedical implication, this application for this case was the importance and challenge of polytherapy and drug combinations, which is motivated by the observation that many patients take multiple drugs at the same time to treat complex diseases or coexisting diseases. It's actually statistics is that 46% of people over 65 years take more than 5 drugs at the same time, prescription drugs to treat those diseases. It's not uncommon to encounter patients that take 20 drugs or more to treat heart diseases, depression, cancer. And that is challenging because patients that take more drugs at the same time are at higher risk of developing unwanted adverse events. 15% of the US population is affected by those adverse events, and that creates a problem both for individuals as for the entire healthcare system in terms of treating and mitigating those adverse events. How, what, and why do those adverse events arise? We're particularly interested in adverse events that arise when a patient takes multiple drugs at the same time. And those events cannot be attributed to either drug alone in a combination. When they take multiple drugs at the same time, and we just have seen that that's common, they are to this higher risk of developing adverse events. What does that mean? Let's look at a hypothetical example. Imagine we have just 2 drugs, a red one and a blue one. So these unexpected interactions between drugs could be described as follows. Imagine a patient takes blue drug alone. When they take blue drug, the desired therapeutic effect is achieved, no side effects occur. When patient takes red drug alone, desired effects are achieved and no side effects occur. However, when a patient takes blue and red pill together, then with some probability, they will develop a particular type of side effects. So that is surprising, that is unexpected, because if the 2 drugs would act independently of each other, we would not expect to see any side effects. So the question then becomes, well, can we have a model, can we design a model that will predict how likely it is that a particular combination of drugs will lead to a particular side effect. And so to tackle this problem, we built a large polytherapy knowledge graph. So that knowledge graph had information for all approved drugs in the US and their known interactions between them and how those drugs work. And so for that first layer of information, where we looked at all approved drugs, each drug is represented as a green triangle, and two drugs are connected by an edge. That edge has a particular type, such as R-sub1 or R-sub2, and the type of that edge is a specific type of drug interaction. So R-sub1 might represent, for example, bradycardia, which would be then interpreted as bradycardia is a side effect that can occur with certain probability in patients that take both drugs together and cannot be attributed to either drug alone in that combination. So we built that knowledge graph together with collaborators from FDA based on their National Adverse Event Reporting System, and we enriched that information with information about how mechanisms of with information about drug mechanisms, in particular their targets and how those target proteins interact with each other. And so this gave rise to this polytherapy knowledge graph. Once we had this knowledge graph built, we developed an algorithm that can learn over this knowledge graph. And so that was a graph neural network algorithm. The way this algorithm worked was that it took each and every node from this knowledge graph, so each protein node and each drug node, and the graph neural network is a nonlinear mapping function F, that has some parameters that we will need to optimize during the process of training. But the idea is that for two nodes that have similar local neighborhoods in the input graph, they will be represented by points that are close to each other in the embedding space. And so this function F really maps nodes to the d-dimensional embedding space, and it does so in such a way that the structure of that low-dimensional embedding space reflects the topology of the input graph. So that if we take these learned embedding vectors and we perform linear algebra operations with them, that will reflect similarity and structure of the input graph. The way this can be done in function F is by specifying some graph neural architecture, which is effectively some form of a neural message passing mechanism that learns what is the right, most effective way of upweighting, downweighting direct and indirect interactors for each node, and send some neural messages along edges of the graph and transforming those messages in a nonlinear way to produce embeddings for each and every node. So broader applications of that work in terms of graph neural networks is that the way you can think of that is that with graph neural networks, they now allow us to apply these deep learning algorithms much more broadly than what was possible before. So we already know that these models work, the deep learning models work really, really well with medical images, where convolutional neural networks transformers are powerful architectures, or with biological sequences using LSTMs and other architectures. But now we can really train deep learning models for any interconnected graph structured data set. And so this creates these new opportunities in biology and medicine, such as this one on polytherapy prediction that I mentioned. So we trained a decagon such that it can answer queries about drug interactions. For example, one might ask a query, how likely a form of statin and ciprofloxacin when taken together would lead to breakdown of muscle tissue. And when that query is asked by a user in the backend, the model will return the probability of an edge of type breakdown of muscle tissue existing between the two drugs that appeared in the query. So that's how the model was trained. And once we trained the model, we did a variety of different sanity checks and performance evaluations. I will not go into all of them. Just one highlight is is a challenging setup known as retrospective data split or temporal data split. Well, we took all data on interactions between drugs that was available prior to 2012. We trained the model on that data. Then we asked the model to make predictions that the model is most confident about and see what predictions have been confirmed after 2012. And so this table is showing top 10 predictions that the model was most confident for, and we found that for many of those we were able to find evidence of patients that were on a set of drugs that were predicted by the algorithm and experienced the undesired side effects later on. And kind of that was exciting. It encouraged us to engage in follow-up evaluations of model predictions that we have now done with a couple of hospitals on the West Coast and also here on the East Coast with Massachusetts General, and collaboration also with Tianxi Kai, where we are evaluating predictions about these new polypharmacy side effects against standard drug interaction markers, lab values, other surrogates in EHR, focusing on drugs used to treat rheumatoid arthritis and IBD. And the results are kind of promising, and we see that there are these novel side effects that the model can correctly prioritize. So where do we go from here? So since this initial work on Decagon and kind of its various follow-ups, we were discussing about like how can we think about this modeling in a more broader way. And so the way we are thinking of it now is that we really have this toolbox on deep graph representation learning that can embed effectively a very large knowledge graphs that are very diverse, might have different types of nodes and different types of edges. And once we have those embedding spaces, we can leverage those spaces to decode novel relationships, novel edges that have not been known before, and make predictions about those novel relationships. And that's possible to do in a more general way, so that we don't only train the model to answer queries about adverse events of drugs, but might be trained, we can decode those embedding spaces to answer different kinds of queries, such as, for example, will a compound A target gene C that in turn participates in a particular pathway D? Or will a particular compound treat a particular disease that, and will it do so by targeting the gene that is expressed in a given tissue? So all those very different kinds of queries can be answered by the same foundational model. So to make some of this work a bit more practical, we recently been working quite a bit on developing these geometric deep learning predictors for modeling therapeutic indications. And so we created a precision therapeutics knowledge graph, which is a very rich heterogeneous graph that contains information about not only genes and drugs, but information about genes and how they are targeted by drugs, phenotypes that those drugs are associated with, how they relate to diseases, exposure, and then biological processes, functions, and cellular components. And then given that therapeutics knowledge graph, we developed a deep learning predictor that can identify what are then and can answer different kinds of queries that I showed on the previous slide. For example, one query that we might be particularly interested in are queries that would identify whether a particular drug might be efficacious for a particular disease. And so this, on the right part of the slide, I'm showing the results of such a model, which we refer to as RxGNN, relative to standard models that don't have the ability to really leverage full rich knowledge graphs. And we can see in particular that the lift in performance measured as area under precision recall curve is particularly prominent in cases where we are interested in prioritizing post-candidate treatments for diseases that have otherwise very small number of treatments or limited molecular understanding. And so that's, I think, also where AI and ML methods should focus more, because that's where they can provide most actionable hypotheses and guide biomedical discovery. And so once those predictions are made, the question is, well, not only should predictions be made, but if you really want to experimentally verify them with collaborators, it's important to move away from just providing point-wise estimates about how well a particular model behaves, but also, well, what is the data that the model relied on when making a particular prediction? And so for that, we've been developing post hoc algorithms that can take any classifier and can explain predictions made by that classifier, and can do so by identifying what are the small number of nodes in a very large knowledge graphs that, if removed from the data, would change model predictions considerably. So that would indicate that those small number of nodes are really what the model is focusing on, and model thinks of being crucially important when making the prediction. And that can be visualized as a form of an explanation. So here in this line, I really want to highlight a very exciting collaboration that I've been fortunate to have with Niels Gellenborg's lab and one of his students, where we were able to connect outputs by these deep learning models and explanations they can provide together with visual interfaces, and really design an exciting visual interface that allows domain experts, scientists, to interact with model predictions and explore different explanations. And
Great to interrupt, Marinka.
Yes.
We're just running out of time. So if you don't mind wrapping up, that would be great. So have some time for questions and answers as well.
Just 2 more slides.
Okay, great, thank you.
And the role of this visual interfaces has been particularly important to kind of do this last mile of research, where we don't want to only develop models and put them on GitHub or put them and describe them in the paper and supplementary material, but really see how they can be more practically useful and to domain scientists. And in this particular example, I'm showing results of a study where we show that by providing predictions and together with explanations that our model can provide, then domain scientists can better understand those predictions. They can, these explanations help them assess the predicted drugs and whether they would trust those novel predictions. And so that concludes this second part of the talk. And for the last one, I just have one shout out, which is that we're excited about designing, creating kind of flagship AI ML data sets for particular focus on therapeutic domain. And towards that end, we have an open science initiative called Therapeutics Data Commons, which is a std commons.ai that I highly encourage everyone to check out, and happy to talk more about it. With that, I'll stop here and see if there are any questions that I can take.
Thank you so much, Marinka. A little round of applause. So with that, are there any questions from the audience? Please feel free to raise your hand or type it in the chat. I see applause. No questions raised. So,
Marinka, sorry, I'll ask, I have one here. So one of the sort of interesting things that happens in sort of what's it called, treatment-resistant hypertension, right, is that people end up on multiple treatments, right? So they'll end up on 2 or 3 more treatments for the same condition. So it's a little different than the model that you had. And I just wondered whether you've thought about, you know, is there ways to sort of think about using that same approach to identify when multiple treatments for the same condition are actually not likely to be synergistic in any way or even additive?
Yeah, that's a great question. That is actually the study of one of the follow-up, the key question of one of the follow-up studies, where I work together with Tianxi Kai and her team at the VA and Mass General, and we are looking specifically at triple therapies for our rheumatoid arthritis and IBD. So for rheumatoid arthritis in particular, there is a fairly common therapy that consists of 3 distinct drugs with different mechanisms of actions. that is prescribed to patients with RA. And so we were interested there in predicting adverse events that might be associated with those triple therapies. And where we currently stand is that we are evaluating predictions against electronic health record data at Mass General, where we have queried patients that are diagnosed with RA and are on those triple therapies and the kind of phenotypes that they are reporting. And we are correlating those phenotypes with side effects that were predicted by the model. And again, there are lots of confounders and biases that need to be addressed in that way. So it's not as trivial as just querying the database and doing the comparison. But yeah, it's an important question that we have in mind.
I get, maybe I misunderstood. I'm a bit less worried about the adverse events. I'm a bit more worried about taking a drug when you don't need to, right? So it has its own problems.
Yes.
And so just wondering if
that's a great question, an important problem which we have not tackled it yet. But it's a problem in the sense of most drugs are just being unprescribed because of patients in so many different specialists, and each specialist will just add a new drug. They will not remove a drug that was prescribed by other specialists. We are not yet tackling that.
Yeah, that's a big problem because we just keep prescribing up, right? And often people have a big treatment regimen for which there's very little evidence that the bulk of it is doing a thing for them apart from potentially making them sick, as opposed to healthy. Right. Great, thanks so much. I have to run to another phone call, but really appreciate you coming, Marinka.
Thank you for inviting me.
Are there any other questions? If not, I have one, perhaps a quick one. Okay, well, I was wondering about the definition of when you're looking at particular subgraphs in your knowledge graphs or in general in your graph representation. When that is defined, these local subgraphs, does it take into account the fact that this knowledge is coming, say, from an ontology? Say you have a cell ontology behind that, where the relationships of the edges are well defined, they have their well-defined semantics, and you can actually infer stuff from them. So in particular, in the ontology world, there's always the notion of here's the graph as it's stated, as the ontology has been built, but we can always infer, there's an inferred representation of that ontology that we can look at. And I was wondering whether that's taken into account in the learning process, whether it's just you take it as it is stated, or whether you do some inference, and what kind of inference, because that might in the end change the neighborhood of a particular node to have perhaps a few more nodes or a few more edges to it. So I was just wondering about that.
Yeah, Rafa, that's a great question. So indeed, we are doing that. So my answer has two parts. One is first is that edges or relationships in these ontologies and knowledge graphs are typically typed, meaning that we have it's not just an edge that would indicate there is some kind of relationship, but there is an edge that has an association type to it, so that we know whether a particular edge has been derived from an experimental, from an experiment, or from a computational resource, or if that edge in the context of cell ontology indicates an is-a relationship or part of relationship. Depending on these edge types, then graph neural networks have a separate processing channel for each edge type. So each edge type will have its own subset of weight matrices that then allow us to allow the model to downweight certain edge types, upweight other edge types if they are more useful for making predictions. The second part of the talk is that by definition, the embedding-based models assume that the underlying graphs that we work with are incomplete and noisy, meaning that we have limited information about them. So there might be false positives in the networks. We might have edges between nodes that actually are not there, which is not uncommon. For example, if you just take protein interaction network, we have lots of false positives that might be due to limitations of experimental platform. And there might be false negatives. So pairs of nodes that actually interact through a particular type of edge, but just that interaction has not yet been measured, and it's actually part of the inference process. And very often we then sort of the goal of optimization is to train a model that can complete knowledge graph. In machine learning methodology, this is known as knowledge graph completion task, which is essentially a task of taking this noisy incomplete graph and identify missing edges. Now, missing edges can be, for example, novel predictions about protein interactions, or, for example, in the context of cell ontology, at one point we were very excited about data-driven inference of those cell ontologies. And our goal was to build a tool that could help ontology curators decide when to add a new term to cell ontology or to gene ontology. And so essentially the way you would frame that in ML language would be to say, okay, the task here is to predict links between existing terms in cell ontology and this new candidate term that we would like to add, but don't really know or exactly where its optimal position would be in the cell ontology. And in the inference process, we estimate then possible links between this new term, new node, and existing nodes.
I see. That's very interesting. Yeah, I'd be very keen to, I'll probably have a look at the website and find some of these papers, but that knowledge graph completion work or ontology completion work is very much interesting and very much an active, desirable process in practice when people are looking, especially when you're looking at this combination or high volume of data and combination of that with what's already known and represented in these ontologies, you'll usually find that there's quite a bit of gap, especially in things like phenotypes and phenotype representations in, say, the human phenotype ontology. And when you look at the data, you see that there's a little bit more there than is already kind of known and represented in those things. So
absolutely.
That is fascinating.
There are also lots of investigative biases. At some point, I asked a student to essentially correlate major NIH cycles and the diseases that those NIH funding cycles were designed for, how complete those ontologies become a few years after those cycles are ended. And it's like very clear and not unexpected in a way, right? To see that if there will be a major NIH call for various centers focused on specific disease area, it's really how you can see that part of the knowledge graph in the next few years becomes much more complete. Right. And so that creates lots of biases, because now it means that just the density of knowledge somewhere, it does not necessarily indicate any biologically meaningful information. It might simply indicate, well, that's where the current research has been focused for, or that is the kind of data that can be measured with existing biotechnological platforms. So yeah, it's interesting to kind of study these kind of questions and see what kind of biases can arise and how to mitigate them.
Absolutely. Oh, this was fantastic. I don't want to monopolize the conversation, but maybe we can get together another time and chat about more of this stuff, more collaboration opportunities. So with that said, maybe I'll close off the seminar and let everyone go. And then thank you so much for joining us today. And it'd be great to catch up in the future. In the meantime, thank you.
Thank you for inviting me. Enjoy the rest of your day.
Thank you. You too. Have a great day. Thanks, everyone.